Hello, my name is Dr. Brendan Griffin, and today I'm presenting a webinar as part of the SSPC webinar series. The title of my presentation is Poor Drug Solubility is a Matter of Fat, and I'm going to make a case for the benefits of lipid-based formulations to improve oral drug bioavailability. This work has been made possible through the funding support with SSPC, funded through the SFI, and I am based in the School of Pharmacy, University College Cork. So the overall plan for my presentation is to set the scene in terms of what are the latest trends in pharmaceutical research and development, and maybe focusing on the challenges facing the pharmaceutical industry in converting molecules into medicines, which is a key area of my research, to understand what these bottlenecks are and to identify solutions to try and resolve that. We'll talk in more detail on the challenges faced with the poor solubility challenge and basically the limiting factor whereby lots of newer drugs being discovered from drug development programs present challenges in terms of poor solubility. In other words, they're not soluble when ingested orally, and as a result, they're not absorbed and don't work in the body. I'm then going to mention that one enabling technology or bioenabling technology that will enhance bioavailability is lipid-based formulations. I'll make the case for where lipid-based formulations have advantages, and maybe in that process identify some of the limitations of some areas where lipid-based formulations are not suitable. I'll give an overview of some of the more recent research conducted in my lab in terms of focusing on maybe advanced lipid-based formulation design. So lipid-based formulations outside of the conventional classes of lipid-based formulations. And we're going to talk a bit about lipophilic salts, supersaturated systems, and maybe lipid suspensions. Finally, I'm going to finish up on what we need to do in the pharmaceutical industry, both in academia or industrial setting, to improve the guidance for industry on how to develop lipid-based formulations, how to streamline those decision-making processes and improve the chances of commercial success of a particular lipid-based formulation with a lead drug candidate. So my slide here is really just to summarize some of the key factors limiting the pharmaceutical industry as we transition from exciting drug discovery candidate to a licensed medicine. So that process of developing licensed medicines can take anything from a number of years up to a decade and more. And what the Nature Drug Discovery Review article by Scannell and colleagues identified that the reverse of Moore's law was at play when we're talking about discovering new medicines. And it proved that basically the number of new medicines approved per investment has been reduced by 50% every 9 years since the mid-50s. So this is the opposite of what you might expect in terms of compared to the microelectronics industry, where increased investment is leading to less licensed products in the market. And there are many reasons for this, including poor efficacy and so on and so forth, increasing costs of clinical trials. One of the problems is increasingly associated with the challenges associated with delivering drug candidates with poor solubility. And we've discussed this before, and we've talked about this in the in the various literature in terms of the potential, and here I've discussed previously in an advanced drug review about the potential of lipid-based formulations. To bring you up to date more, in the last couple of years, increasingly we're seeing drug candidates emerging from drug discovery programs that are maybe outside of the scope of the traditional drug-like properties that we would have looked at. So Lipinski's rules, for example, would have said drug-like filters and guidances and molecular weight log P and so on and so forth. And over the last 2 or 3 decades, these have been a good guide to how to develop good drug candidates with good developability characteristics. However, what we're finding now is that there's more and more products that are outside of these classic drug-like filters, or outside of these classic beyond rule of 5 space. And increasingly, as illustrated in this particular paper by Michael Schultz, that the average molecular weight of new drugs licensed by the FDA exceeded 500 for the first time in 2018. So we're getting more drugs coming out of drug discovery programs that are not less ideal in terms of developability characteristics. They have properties outside of the ideal properties for developing medicines. And this is presenting challenges from formulation because these products are even more difficult to deliver, displaying extremely poor solubility. So, for example, you're seeing products being brought to the market which are basically in poor formulation types and so on. So they're presenting challenges. These are creaky formulations, as has been coined by Henbrick and their colleagues. In addition, I just want to set the scene in terms of where we're at over the last couple of years in terms of increasingly an accelerated access to market R&D model. So facilitated by regulatory agencies or the prime or priority medicines initiatives, companies are increasingly expediting and accelerating their drug development programs to meet priority medicines needs. So you're getting lots of more products being brought to the market in a shorter time frame. And I suppose there's less time to make key decisions in terms of formulation, optimizing formulation, and selecting the right formulation for clinical trials. So drug development and formulation development scientists are working in a particularly narrow space where we need to get these products on the market as quickly as possible, and we want to take a lot of the guesswork out of that process of developing new medicines. So these are kind of some noteworthy things that the vast majority of products licensed in 2018 were orphan designation products. So clearly these products are rushed to the market for relatively smaller patient population cohorts, and key information regarding formulation and optimizing the formulation to achieve the optimum for the patient in some cases can be lacking. So try and put that in the perspective of what this means for the patient. I've mentioned this high-tech drugs in creaky formulations as an example that we're rushing these formulation development to the market. But here's some studies we've shown that basically many of these products are coming to the market and they haven't been optimized for patient use. So, for example, food effect and understanding variability as a result of taking your medicines after a meal, this can present significant challenges for a patient. So, in a recent review article, we've identified that over 40% of the newly licensed drugs over the last number of years display significant food effects. So, this presents significant challenges for patients because they have to follow strict labeling instructions, whether they should take it with food or in the absence of food. And this, in terms of the modern flexibility that we have in our lives, presents challenges to the patient to maintain that consistent prandial conditions for allowing it. So as an example, you can see products here such as Orkambi was licensed a number of years ago. In fact, the product information tells the patient that you need to take the food with a fat-containing meal or snack. And they give advice, since the FDA product labeling says you should take it with cheese, whole milk, whole dairy products, meats, and oily fish. So this presents new challenges in terms of guiding. Here we have a regulatory agency actually advising what food you should take or what specific types of food. And you have to ask the question as formulation scientists, did we really perfect that formulation? Do we have to use food supplements or food to give that bioenhancing effect? So just to illustrate even in more extreme examples, this is the information provided. So classically, product information provided to patients is usually dosing conditions, advice on what to do in specific patient populations. But here we have a particular website that gives guidance on how to cook the right meals, or at least a recipes for high fat containing food as part of the product information or medical literature. So this really hammers home the kind of challenges that we're bringing lots of products to the market. We're not optimizing them for consistent absorption. And in fact, we're getting to the stage where companies are advertising recipes on their product information literature to try and guide. So this is kind of the backstop of how we have to try and address these challenges of formulation scientists. So I call this the kind of balance between good science and patient compliance. So for example, here's another product, Ticabri, is a product that's can be taken. You get 3 to 4-fold higher bioavailability when you take it when you administer it with food. So the product has a significant food effect, and it's an anti-cancer compound. However, the FDA licensing actually advises to take it on an empty stomach. So Despite the fact that it has a significant food effect, the FDA or the regulators advising take it on an empty stomach. Why is that the case? Most likely because it eliminates the variability, because different patients will have different meals might have different impacts, so you're going to get lots more unpredictable clinical responses. So in that case, given that we don't have a thorough understanding of how to enhance bioavailability, the best option is to keep it consistent and go with the lowest amount. So this creates significant losses, significant challenges, and you can see kind of headlines like this: you're flushing oral oncology drugs down the toilet, essentially, by trying to achieve a consistent profile. We're not optimizing delivery, and we're producing issues like this where you're getting headlines to say that we're flushing down patients and payers good funding down the toilet because of those results. So that's the backstop. That's the backstop to my formulation or discussion. I wanted to kind of discuss why we need optimizing formulations. Now I'm going to go into kind of discussing in more detail the challenges facing drug development in terms of addressing solubility. So most people who would have been dialing into this or understanding that why formulation is important would be aware of the poor solubility and the need for bioenabling formulations. And there are many different formulations that have been proposed and identified as commercially suitable formulations. Lipid-based formulations have been around for a number of years. Co-crystals more recently, and amorphous solid dispersions are also a well-established Sporanox and Keltra. These are commercial platforms. So there are many commercial products in the market with lipid-based formulations and amorphous solid dispersions. These represent, but there are other options out there in terms of getting your poor solubility drug candidate into a suitable bioenabling formulation for commercial launch. Now, it's also as well important to kind of illustrate that when we talk about poor solubility, there are some aspects about solubility that need to be discussed. So when we talk about solubility, there are the schematic here presented from Bergstrom Advanced Drug Delivery Review article highlights some of the challenges, the various steps in terms of understanding the transition from a solid state to a solution state. And there are three key entropy changes or enthalpy changes that occur, where you have essentially break up the crystal lattice, and then you have replacement of that space and surrounding of solvation, and then you have an energy gain as a result of solvation of that particular drug molecule by the surrounding solvated solvent molecule. So you can have three stages of this process, and invariably the middle stage here in terms of integrating the molecule within the space provided by the solvent models generally doesn't contribute. But the two key contributors to energy is the breaking up of the crystal lattice, as illustrated here, the delta G, and that's highly predicted by the melting point. So essentially it's a measure of how tightly the crystal lattice binds together. And at the other extreme, you can have generation of energy, so solvation. So basically, if the molecule likes the interactions it makes with the solvent molecules, it generates an energy gain, and you can have a favorable aspects in terms of solubility. So essentially, between these two extremes of solubility steps, you can have essentially solid state limited, where the product is so poorly soluble that the crystal form is so much energy is required to break up that crystal lattice. You have a solid state limited solubility characteristic, and in the literature we tend to refer to these as brick dust molecules. So essentially they're very, very tightly bound, and you get products that are high melting point and that aspect. And the other stage, you might have products which are solvation limited, and essentially they're poorly soluble. They're highly lipophilic, high log P. So we would refer to these as solvation limited, or what classically we refer to them as grease ball type molecules. So in the case of brick dust versus grease ball, both are poorly soluble. In one case, however, is the melting point or crystal lattice is the primary limiting factor for solubility, whereas in the latter case of the grease ball molecules, it's the ability to solvate with the solvent at the end is the limiting factor, and that keeps solubility low. So understanding solubility outside of the overall level, and understanding the limiting factors, whether it's melting point or log P, and the balance between these two, can have a significant impact in terms of designing the right formulation approach to address these particular challenges. So in terms of what bioenabling formulations can be done to achieve this, so as illustrated in the schematic here, you can see classic dissolution studies in conventional dosage forms have a typical profile like this. Now lipid-based formulations, and in most cases what we refer to as self-emulsifying drug delivery systems, generally form microemulsions or swollen micelles in the gastrointestinal tract. So these are mixes of lipids and surfactants that self-emulsify and form emulsions or microemulsions within the intestinal, and during this process the drug is maintained in a solubilized state. So the key advantage of lipid-based formulations is that the drug is dissolved within the lipid excipients. So what we refer to this within the literature, we bypasses the classic dissolution step. So by being soluble in the lipids, it's in a molecular dispersed state within the lipids, and then it transitions into a microemulsion or aqueous micellar system to generate sufficient drug solubility within the gastrointestinal fluid. So we talk about this being solubilized or maintaining solubilization on transit through the gastrointestinal tract. This is in contrast now with classic amorphous solid dispersions, which must undergo a dissolution process, and that can be a critical factor governing how much of the drug is ultimately presents into solution. But in an ASD format, the polymeric matrix supports the rapid disintegration and generation of a supersaturated state. So not to go into too much detail, I mean, ultimately we talk about a spring and parachute approach. So with amorphous solid dispersions, classically we describe that once it's in this amorphous state, it generates this spring or propensity to supersaturate on dispersion in aqueous media. And then we can have a parachute phase where it prolongs that period of supersaturation. And classically, the selection of the polymer and the crystal form can generate or determine the rates and kinetics of both the spring and the parachute effect. In the context of lipid-based formulations, we also refer to these in terms of supersaturation and spring and parachute effect. Slightly different understanding, however, because ultimately the drug was originally starting with a high solubility because it's originally dispersed or solubilized within lipids. But you can get this supersaturation phase where the drug is maintained above its equilibrium solubility as a result of the transitioning processes, and in some cases digestion can lead to that. So an understanding of the supersaturation and precipitation, or the balance between supersaturation and precipitation, can provide a useful insight into which formulations can be suitable for enhancing oil availability. So in terms of the case for lipid-based formulations, I'm just going to summarize really why lipid-based formulations are reported or have been shown to enhance oral bioavailability. So when we talk about lipid-based formulations, classically we're talking about gelatin liquid-filled capsules, which is a lipid containing surfactants in a liquid state, containing the drug solubilized. When this mixes and dilutes in water and gastrointestinal fluid, generally the ones we look at are self-emulsifying. So they form emulsions with low interfacial energy as a result of the surfactant mix that's present within the formulation. So the drug in this case is maintained in a solubilized state. And this process of maintaining this by using the right surfactant mix, this self-emulsifying promotes solubilization and facilitates integration of the formulation lipids into the natural dietary lipids that exist in our gastrointestinal tract, which are bile salts and phospholipids. So you get an ease of assimilation, readily assimilation into this pre-absorptive bile salt mixed micellar milieu that exists in our intestine and absorptive. And this is the actual conduit by which drugs get carried. So essentially the bile salt micelles play a key physiological role in terms of absorption of lipophilic nutrients, and in many cases for poorly soluble drugs, it's the incorporation of the drug within the bile salt micelles that facilitates uptake in the gastrointestinal tract. So by promoting solubilization, by generating solubilization, which could be as a result of supersaturation in a post-digestive state, this facilitates the permeation or the flux or the transfer of drugs into the systemic circulation. The second major mechanism by which lipid and lipid surfactants function in terms of a biopharmaceutical sense is that many lipids and surfactants are known to enhance permeation, and by that I mean they increase flux across the enterocyte membrane. Now the exact mechanisms involved are quite various, and they have been widely discussed within literature in terms of whether they increase paracellular through a tight junction opening, or by increasing permeability through the membrane flux, or by altering PGP. So while there's many reports in the literature, depending on the types of lipid, overall, it is generally universally recognized that lipid excipients themselves exert our natural permeation effect. And again, beyond the scope of what I'm going to talk about today, but this is another significant advantage to lipid-based formulations. The final mention that I'll refer to in lipid-based formulations is the ability to stimulate lymphatic transport. So what is lymphatic transport? All of our dietary lipids, our fats or oils that we ingest in our normal diet are not directly transported into the bloodstream. Most of them are transported by chylomicrons or triglyceride-rich lipoproteins that are synthesized in the intestine, and this is our transport mechanism by which we ingest most of our fats. So in some cases, lipid excipients can stimulate this lipoprotein production, and that process can actually lead to carrying drug through the lymphatic system, and this can be a very effective way as well by which lipid-based formulations can stimulate lipophilic drug uptake from the gastrointestinal tract. So these are the various mechanisms by which lipid-based formulations stimulate and enhance bioavailability. As an example, this is a study by Les Bennett and colleagues just to illustrate how lipid excipients or common food dietary lipids that we see in our diet, such as oleic acid, can be used to reverse efflux mediated as a result of transporters. So by using oleic acid, you can reverse the efflux observed as a result of PGP mediated efflux on this particular substance here, where in MDCK1 cells, by exposing them to oleic acid, you reduce vinblastine efflux. And again, these are natural lipids functioning that are present in all of our diet. And while it's still unclear exactly what is the physiological impact of these, we've demonstrated, or it has been demonstrated mechanistically, that common lipid excipients function as, in this case, reducing efflux and therefore increasing permeability through the gastrointestinal tract. So there are numerous examples. So it is a well established commercially viable platform since really the flagship product was back in the early 90s with Neoral or cyclosporin, and this really transformed the way by which at least the industry viewed the concept of commercializing a soft gelatin capsule containing a lipid mix with the drug dissolved in this, and you can see the advantages of them. So there's many cases, and also there's a wide array of lipid excipients utilized to do this, and there are some classification schemes. So there is an LFCS or lipid formulation classification scheme, and they tend to group them into 4 categories. Again, beyond the scope of this, but if you look up the kind of LFCS system proposed by Colin Poughton and his collaborators down in Monash, they've done a lot of work in terms of demonstrating a classification scheme for lipid-based formulations and guiding that type of concept. Now, to give some examples of some of the work we are doing here in the School of Pharmacy, UCC, we've shown for a number of years now the benefits of lipid-based formulations. So one of the model compounds we've looked at quite a lot is phenofibrate. It's a high log P compound, and when the product was brought to the market, initially it was brought to market as a micronized Lipantal micro product, as you can see here, and this product has a food effect labeling. So the patients must take Lipantal micro after food to get the maximum benefit, and that presents significant challenges. What we've done is we've reformulated that with a lipid-based formulations, and we've demonstrated by formulating with lipid-based formulations you can bridge that gap between fed and fasted state is how we coin it. And really, to illustrate that here, what you see on this slide, in the fasted state with the lipid-based formulation, you get very high concentrations in the beginning and all of the drug gets absorbed. In this study in pigs, however, what you see with the micronized product is when you give the drug for the same dose, you don't get the same amount absorbed, and in fact many of it still stays in the intestine, so isn't fully dissolved. And it's only in that you get a secondary absorptive phase, or what we refer to as a secondary peak. After you've taken your dose on a fasted state and then you have some food a number of hours later, that food stimulates the production of endogenous bile salts and so on, and that process actually stimulates increased solubilization of the Lipantal microproduct, and you get absorption. So that's the classic micronized product. When you formulate as a lipid-based formulations, you immediately simulate that postprandial conditions by using lipids in the fasted state to bridge that gap, to stimulate those those bile salts effects that you need to be able to enhance bioavailability. And you can see that lipid-based formulations would be used for this. We've also benchmarked processes. So for example, we've compared a nano formulation, a Lipantal nano size. We've looked at silica-based formulations. We've looked at many different formulations. In all of our studies, lipid-based formulations, at least in our cases with this particular molecule, lipid-based formulations display the highest bioenhancing effect. And again, I should caution this is particularly one particular drug, but it shows that you can demonstrate a bioenhancing effect with lipid-based formulations, and in this case for bridging a food effect gap, we've shown and we've published on. So the as a commercial platform, lipid-based formulations are significantly, they've a proven track record. However, we've recently published and we've looked at over the last, say, 5 or 6 decades, how many lipid-based formulations on the market. So what you could see in the 80s and 90s, there was quite a lot of lipid-based formulations coming on the market, but it seems to have peaked in the 2000s, and in fact, in more recent times, it seems to be losing ground, certainly relative to solid dispersion formulations. So while it's well established in the 80s and 90s, there seems to be some reason why lipid-based formulations are less common or less familiar in recent times. And I just want to explore some of the reasons for these kind of factors. Now, part of it is that it's not absolutely clear, the various types of systems out there. In many cases, there are lots of questions still out there in terms of understanding which is the right type of lipid-based formulations, and there are challenges relating in vitro observations to in vivo, so these can be challenging, and I refer anybody to look at papers that might explain that in vitro in vivo correlation for lipid-based formulations. But also I just want to discuss and to capture one of the points that I've mentioned earlier on in terms of the types of drugs that are coming on the market. So these drugs that are in the beyond rule of 5 space. So increasingly we're seeing an increasing prevalence of drugs with a higher melting point coming on the market. So these are the brick dust molecules, and these molecules are inherently poorly soluble both in water but also in poorly soluble in lipids. And the problem with these kind of brick dust molecules is they have neither solubility in water, but they're also very poorly soluble in lipid formulations. So if you can't dissolve in the lipids, you can't formulate a lipid-based formulation. If you can't formulate it as that, the big advantage of bypassing dissolution is negated. So we don't achieve what we need to do. So the dose isn't soluble because they have a higher melting point and because they are inherently poorly soluble in lipids as well as water. So these brick dust molecules do not readily assimilate or fit into these kind of SNEDDS or these types of systems here. So we've got to look at different strategies of how we can overcome these brick dust molecules, or at least overcome the limitation for brick dust molecules being loaded into lipid-based formulations. So this is some of the challenges that we're going to discuss in some detail in terms of how we can look at ways of advancing and exploring opportunities. So here's some studies we've looked at in terms of lipid suspensions. So what are lipid suspensions? Well, basically you're adding in drug above its equilibrium solubility in the lipids. So essentially what we've shown is that in this case, here's a Nilotinib, a classic newer compound licensed in recent years, which is a beyond rule of 5 space, very poor solubility in lipids, and here we've shown that when you use the formulations as lipid suspension, you get higher bioavailability. But more importantly, when we refer to a chase dosing, so in this case you give the suspended drug, but you also just follow it on or chase it on with lipids such as a placebo lipid formulation, so the drug is not inside because it's not simply soluble. We've shown that you can get further enhancements in bioavailability using this chase dosing or lipid-based formulation approaches. Another example is we've published work where we've found a 5-fold increase in bioavailability with selected surfactant types. So for example, using the same model compound, Nilotinib, we were able to increase bioavailability fivefold using a certain types of surfactants. So again, tween 20 was the selected one here, and we were showing that pure tween 20 based lipid based formulations could overcome bioavailability. So the commercial product, or this commercial product here in our control study, was an aqueous suspension, but we can enhance exposure fivefold. So a significant improvement by using tween 80. Other options you can look at for lipid based formulations are the use of supersaturated systems, and in this case we use heat to increase the amount of drug dose loading above the equilibrium solubility. So again, using supersaturated lipid-based formulations to get higher drug concentrations than can typically be achieved at room temperature. We've looked at this with another compound called Venetoclax, another beyond rule of 5 space with a very large molecular weight. Commercially, the product has a significant fed to fasted effect. So Venclixto is the commercial product, fasted and fed state about a 3-fold higher bioavailability. Whereas when we look at a supersaturated system, we can achieve the same level of bioavailability that you would achieve in the fed state. So again, what we're doing is harnessing the benefits of lipids to mimic the fed state conditions. And this formulation achieves significantly higher than both the fasted state and the pure crude drug substance. Another strategy we've been exploring is the use of lipophilic salts. So here, as I've mentioned, for many of these drugs which are more challenging to deliver, they have poor inherent solubility in lipids, so we can't get a sufficient dose loading. So what we've done is try to redesign the salt form. Rather than a salt that actually enhances aqueous solubility, we want a salt that enhances lipid solubility. And we looked at 3 different strategies by looking at different counter ions. For our work in our particular study with Venetoclax, it didn't work with oleic acid or decanoic acid, but it worked quite well with docosite, which we can see here. And so what we were able to demonstrate in this is, and again using the lipophilic docosite salts, we were able to reformulate this product to generate a lipid-based formulation with a high dose loading, and again being able to match, and in some cases maybe even exceed, the fed state commercial product. So the commercial product out there has a profound food effect, up to 3-fold in pigs, in our study up to 5-fold in humans. Using a lipophilic salts in a lipid-based formulation, we were able to match that in a fasted state. So again, you can see the benefits of lipids bridging that and harnessing the inherent benefits of lipid excipients. Another case I just want to mention is the use of precipitation inhibitors. We've also looked at incorporating precipitation inhibitors that prolong supersaturation in supersaturated lipid-based systems. Because they're kinetically inherently more unstable because you've generated a supersaturated state, we can look at strategies to include a precipitation inhibitors. And you're getting to even more complex combination formulations in this case. And in this case, we used a supersaturated lipid system with a precipitation inhibitors to enhance exposure, here using poloxamol 407 to achieve this. So again, this is just a snapshot of illustrating how we can use lipids to enhance exposure and maybe also use precipitation inhibitors. So to summarize then, where would we go in terms of what we're finding about lipid-based formulations? And part of our work is to try and provide new guides on which, rather than trying every type of formulation strategy, we need to provide guidance on which ones are best matches depending on your drug properties. So this is really where working within a consortium referred to as PEARRLO, which I'll provide some information, we look at strategies that would provide industry's decision trees. And again, beyond the scope of this particular study, but it gives illustrations where, if you have the ideal characteristics, you should look at a conventional dosage form and doesn't need to be any more complicated. If you have products which have a good dose loading that form solutions readily in a classic SED system, we'd recommend going this. However, if you need advanced lipid-based formulations such as products which have a high log P, a high melting point on those brick dust category, we've got to look at the other types of lipid-based formulations such as lipophilic salts or surfactant-based systems, or maybe a combination product such as this. And these are kind of examples that are stating these points. So again, beyond the scope, but we've provided further details within our published reports within PEARRLO to kind of how to rationalize those decisions and give industry better guidance. Rather than doing a test on every single lipid-based formulation, can you kind of select a priori which one would be best based on your drug properties? Finally, within that guide of trying to provide better insight into which lipid-based formulation is the best match for a particular drug substance, we've been looking at new computational pharmaceutics approaches to try and improve those decisions and using computational tools to be able to link those. So by looking at kind of solubility measurements and high throughput mechanisms, trying to see what solubility gains or solubilization gains you get with lipid-based formulations, linking those to specific drug properties and molecular properties, and we've published on this, and we have now a paper in press on this. And essentially what this is, we want to generate predictive equations, and in this case is based on log D, number of aromatic bonds, and melting point. This will give an idea that in the discovery stage, if you know your melting point, you know the number of aromatic bonds, and you know your log D, you can predict what the solubility gain would be for that particular drug candidate. So it's a very powerful computational tool to be able to predict, will a lipid-based formulation be a good source for delivering your dose? And this can be extended in terms of the concept of trying to understand the dose and the developability class. So essentially what we want to use this predictive computational tool to consider your drug as a poorly soluble, maybe DCS class 2B compound, applying a computational approach based on its molecular or physiochemical properties to see, would you move it in or would you transition into a more higher solubility category, such as a DCS class 1. So this is just another area where we can provide improved industry guidance using computational tools to predict if a lipid-based formulation is likely to be successful in terms of enhancing availability. So in terms of conclusions, I've kind of given a slide here to try and illustrate some of the decision trees that we've looked at. Really what I want to summarize is that lipid-based formulations have a number of bioenhancing effects in terms of solubilization, enhancing permeability, so significant opportunities for delivering poorly soluble drugs, and they've demonstrated commercial successes over the years. However, there are challenges facing the industry in adopting this. We need better guidance, we need better decision trees, and we have to be conscious that the newer classes of drugs don't readily assimilate into classic SEDDS and types of systems. So we've got to come up with better ways, because lipids are an endogenous, are physiologically relevant, and in many cases have a bioenhancing effect. So we're aware of the benefits of food for enhancing bioavailability. So we just need to try and harness those understandings and to be able to maximize them by incorporating these lipid or lipid surfactant systems into our oral dosage forms. And finally, we need to look better at computational skill sets or computational tools to be able to guide the pharmaceutical industry on which are the right drugs for a particular formulation and give guidance on these categories. So these are just some summaries and thoughts I'd like to share with you in terms of lipid-based formulations. I hope I've given a summary of why lipid-based formulations have advantages. And I'd like to also acknowledge my various collaborators over the number of years who've helped in terms of guiding this. So as you can see up here, many of these are our colleagues, academic colleagues and past PhD students. I've also just referred to the PEARRLO network, which is a Marie Curie ITN, which I've coordinated over the last number of years, and it's built up a strong network of experts in the field and provided us the opportunity of benchmarking, for example, lipid-based formulations versus alternative technologies such as solid dispersions. So we've published quite a bit in terms of the industrial guidance on how to streamline those decisions to ensure that products get to the market quicker in terms of formulation. And finally, our funding agencies, which are listed here. So with that, I'd like to thank you for your time, and I